Today we will discuss transcription elongation in eukaryotic cells. This is a critical phase in which the information in DNA is copied into RNA. Transcription starts when the RNA polymerase, an enzyme, binds to a specific DNA region called the promoter. There are multiple RNA polymerases that specialize in transcribing different types of things. In this video, we will focus on RNA polymerase II, which synthesizes all protein coding regions. During transcription elongation of the protein coding regions, RNA polymerase II moves along the DNA template strand, synthesizing RNA by adding nucleotides complementary to the DNA sequence. For example, adenine pairs with uracil and cytosine pairs with guanine. This process happens at a speed of up to 40 nucleotides per second in eukaryotic cells. Transcription elongation in eukaryotes requires more than just RNA polymerase. Helper proteins play critical roles to ensure the process is efficient and accurate. One important group of proteins is the elongation factors. These proteins prevent RNA polymerase from stalling when it encounters obstacles in the DNA. During transcription, RNA polymerase II can encounter obstacles such as DNA damage, mismatched nucleotides, or tightly packed chromatin. These obstacles cause RNA polymerase to pause or backtrack. Helper proteins like TF2S and FACT assist in resolving these issues. TF2S facilitates the removal of errors when backtracking occurs, while FACT modifies chromatin structure to allow RNA polymerase to access the DNA. If RNA polymerase II incorporates the wrong nucleotide, the elongation factor TF2S facilitates the cleavage of the error-containing RNA, allowing RNA polymerase to reverse and transcribe the region correctly. The 5' cap is a special structure added to the beginning of the RNA in our cells. It's like a protective helmet for the RNA. Here's how it's made. First, an enzyme removes a phosphate group from the very beginning of the new RNA. Then, another enzyme adds a molecule called GMP to this spot. Finally, a third enzyme adds a special chemical group to make what scientists call a methylated guanosine. This cap is super important. It helps protect the RNA from being destroyed too quickly and helps the cell read the RNA to make proteins. In eukaryotes, how transcription ends is less well understood than in bacteria. However, it involves cutting the newly made RNA and then adding a string of adenine molecules, a type of building block, to its new end. This process, called polyadenylation, helps protect the RNA and make it more stable so it can be used to produce proteins. The polymerase keeps going a bit further before eventually stopping and letting go. In summary, transcription elongation of protein coding regions involves RNA polymerase II synthesizing RNA, supported by helper proteins like elongation factors. These proteins ensure the process is smooth and accurate. Thank you for listening.